to be here at Sister Nivedita University. Actually, this university actually stays very close to my heart. I frequently come here for different meetings or conferences. Always I love to be here. So again, that pleasure today I am also having. So another very great pleasure is to introduce Professor Chittoranjan Choudhury. He is currently a professor, an academic dean and dissertation lead of the University of Bolton, City of London Dental School. After completing his undergraduate qualification in dentistry in 1980, he did postgraduate diploma in applied nutrition and dietetics from Dhaka University in 1982 and was trained in oral and maxillofacial surgery from Okayama University Dental School, Department of Oral and Maxillofacial Surgery, Japan. In 1991, he obtained his PhD from Asani University School of Dentistry, Department of Oral and Maxillofacial Surgery, Japan, by investigating the immune status of oral cancer patients with relation to retinol, alpha-tocopherol, and zinc, and first quantified zinc therapy to restore immunity in them. He completed MPH from UK University of Manchester and MFDS from Royal College of Physicians and Surgeons, Glasgow. Then he did his postdoctoral training and research in same subject, oral and maxillofacial surgery, and at Nara Medical University, Japan. He has over 25 years of teaching, research, and clinical experience, also guided many PhD and master students. Professor Choudhury was the founding chairman of the Nitya University Department of Oral Biology and Genomics at Nitya University, Mangalore, India. He is the president of the Indian Academy of Oral Biology and past president of the UK Institute of Health Promotion and Education. He established an international fellowship program in OHN Oncology and Reconstruction and PhD degree course at Swaraj Gupta Cancer Hospital at Calcutta, thereby linking with the faculties of UK and Japanese University with Calcutta. Then Professor Choudhury published many important research works and wrote 7 books, including Handbook of Oral Cancer Screening and Education, which is recommended by British Dental Journal. That is actually very praiseworthy. He permanently lives in UK. Above all, Dr. Choudhury is a humanist and cares nature and the wilds passionately. So, not going into details of, then I will go on telling about Dr. Choudhury. So, I will invite Dr. Choudhury. Please, the dais is yours.
This, can you just confirm? Forward this one.
Forward this one.
Okay.
And back this one.
And this one? Pointer. Pointer is not working.
You can only move the slides.
This is forward.
Yes. This one is for backward.
Okay. Thank you very much. Good afternoon, everyone. Yes, I am not to be introduced by myself here because you know about me from Dr. Das here. I thank you very much for a nice introduction about me. And I am son of the soil, no doubt, but currently I am living in UK. But I am very much passionate to develop something for my country, India, and in particular for West Bengal. Calcutta is my base now, and I hope SNU will help develop certain things which is very needful for our country, for our society. Of course, I am a scientist. My base is dentistry, but I am no more dentist, practicing dentist. I am just developed certain things in a way so that I can contribute science and I can contribute research for clinical applications, so that we can do certain things to just prevent and control of cancer. My main focus is cancer research for prevention and control. And I will just let you know about the situation, what we are facing now today in this country. If we say we have almost 200,000 people are suffering, detected with oral cancer in this country, and it's going up day by day. Unfortunately, many of our young generation now today, even during this visit, I have seen a lot of young people, they started gutka chewing, that is panparag, and then khaini things, very dangerous things. I have just done some studies with the khaini chewing tobacco related oral cancer that has been just giving some expression of mutation and deletion of tumor suppressor genes. I'll just give you certain glimpse about that, and which I studied in Japan with my Japanese collaborators at Tokyo Dental College in Tokyo. And that is very disturbing for us. And when I come here, then I still see that a lot of young generation, they are just buying that sachet, that is the tobacco sachet, and they're putting them in their mouth. Not only the working people, even I have seen the literate people, they are doing the same thing. And they are suffering from oral cancer and oral precancer. And many oral precancerous conditions are transforming to cancer. And you'll see there are many young children, young population, that is the young adults, better to say, they started gutka chewing and they developed oral submucosal fibrosis. They cannot open mouth clearly, and they develop certain changes inside their mouth in the oral mucosa, so that changes leads to dysplasia, and different grade of dysplasia leads to cancer. And this is very dangerous things we are suffering from in this country. We are seeing in this country, and it should be stopped, completely stopped. When I was working at NIT University, that is ABC Dental School, dentistry, then I established a NRT clinic and oral cancer screening and education things. And I have also joined certain activist group to stop this chewing tobacco, to cancel. Of course, this government has just introduced new rule not to just smoke tobacco or cigarette or certain things, but they cannot stop these things of chewing tobacco. And it's very dangerous, and it's very, very common in this part of India, especially in West Bengal and Calcutta. A lot of people, they are just smoking and chewing tobacco. That is causing a lot of problem now. So if I can give you some of the glimpse of the patients who are suffering from cancer and precancer, it is disturbing. So we have to stop it. And how this expression of cancer and precancer transforming in depth by changing at molecular level, that is also very disturbing. So several important tumor suppressor genes are deleted and mutated. And we have gene mapping on that, and some important genes also deleted and mutated, of them 3P and 2Q, 21Q. So those things, long arm and short arm, has been deleted. I'll explain to that. Apart from that, I'll just give you some sort of information about the stem cell. Of course, Dr. Seno explained in depth, based on his findings, very impressive and very nice presentation and insight he has given to us. But certain things of my concern of oral cancer regarding the stem cell therapy in case of oral cancer especially. So there are some limitations and some hope. I'll explain few things about that. Another is the AI technology, how we can use this AI technology for prevention and control of cancer. And I am not very much interested for the treatment of cancer. Once you get the cancer, there is no answer. You have to, there are very, very slim hope for survival if a person develops cancer. And there are survival rate is more than 5 years is very, it's not very common. Say that is the problem. Many people, they have to die within 5 years. So how to prevent and control? This is our strategy, it should be. And it's not very expensive that way. You have to involve the community, how to develop this sort of initiative to just incorporate the patients and the community so that they learn how to stop and control cancer by themselves. So those aspects, I will speak some certain things. And little collaborative research with my Japanese colleagues still going on at Yamagata University. That it's not Okayama University, another university, Yamagata University, so that salivary metabolites. And this salivary metabolites things also we're trying to just validate with the regression model so that the salivary metabolites will be used as the biomarker for cancer screening and prognosis. So those things we are also doing, and I'll explain certain things about that. So now, if I start with my presentation with the images, then it will be disturbing. I'm sorry to just show those pictures of the cancer patients and precancerous lesions in a mouth cavity of some of the people suffering from these conditions associated with your chewing tobacco. So, of course, this is lifestyle-induced disease, and we have to tackle oral cancer, a deadly oral health condition, as I explained to you. So, we have some highlights from chewing tobacco and oral cancer. I will explain about action program and ongoing researches on this area of chewing tobacco associated oral cancer. So you can see this lady was a case of oral squamous cell carcinoma and was chewing tobacco for a long time. And this is the condition, and this patient is no more in this world. And the destruction is so much due to the cancer, and the area is extensively destructed with these cancer tissues. So this is one of the pictures we can see that how the chewing tobacco can cause oral squamous cell carcinoma. OSCC means oral squamous cell carcinoma. Of course, to take the picture, we took the consent from the patient. See, this is oral precancerous lesion, erosive and lichen planus type, PLANUS. I'm sorry for a spelling mistake here. So this is also one of the precancerous conditions which may lead to cancer. So we have seen a lot of patients, people are suffering from oral precancerous conditions with erosive and lichen planus, which is just due to tobacco chewing habit. So this is another frank cancer, oral squamous cell carcinoma, and this is very aggressive type of carcinoma, and started at retrocommissural cheek area of that case. And this patient was also associated with the chewing tobacco for a long time. A benign geographic tongue, migrant variety. This condition is not very is not a precancerous condition, but a lot of papilla is just going out from this surface of the tongue, and that may just giving some sort of fungal infections over there, and that is one of the conditions that is very common. Commonly we can see among the patients who are having this chewing tobacco and you're smoking. The black hairy tongue of a smoker and occasional quid chewers, they do have the same problem in this way. And lichen planus, combined atrophic and plague variety, this condition may lead to cancer. And we have seen a lot of patients, they are having this sort of problem or this sort of condition which is associated with the chewing tobacco and smoking. This is extensively, this is very, very much prone to develop cancer, this sort of precancerous condition, very candidal leukoplakia. And it's very common. And it's very difficult to just treat the patients even with the, some of the drugs, and without any surgical intervention possible, we cannot help the patients to revert this condition. So this is also associated with the chewing tobacco and smoking. So we just, with the Swedish one of the universities, Göteborg Technology University, we developed a computer-aided pattern recognition of precancerous lesions. And that helped a lot to just identify the areas which are precancerous condition. Without just taking any biopsy, just taking the picture and then analyzing the picture, we can just see the pattern of that the precancerous condition, and we can diagnose. This is one of the things we have done. This is a case of oral submucosal fibrosis, what we have seen. The patient, this is very common in this country. And even I have seen when I was just living for first few days in this city, a lot of young people, they have developed this, the oral submucosal fibrosis condition, and which is due to the pan chewing habit or gutka or khoini chewing habit. And many of them who are suffering from oral submucosal fibrosis are developing oral cancer, because this submucosal fibrosis is not only causing the restricted mouth opening, it's also involved the nasopharynx, then hypopharynx, then tongue. So they cannot just eat properly, they cannot speak properly, they cannot put to their tongue, they cannot keep clean of their mouth cavity. So, and of course, this condition is dysplastic condition, which may lead to cancer. At least if we take 100 cases of oral submucosal fibrosis, we'll see that 7 to 10 people are developing cancer. So this is one of the conditions which is associated with the pan chewing, gutka chewing, and smoking at the same time. And of course, the synergistic effect of alcohol drinking, creating this sort of things very severely. This is homogeneous leukoplakia, but this is not very much common condition to develop, to transform into oral squamous cell carcinoma. But we have seen that 2 to 3% of this homogeneous leukoplakia may develop cancer, but which is heterogenic, that is very disturbing for us, and it is developing cancer for that heterogeneous type of the leukoplakia. This is also non-homogeneous leukoplakia, which I told you. This area, the cracked area in the cheek, this is also due to the pan chewing habit, khoini chewing, and smoking. And this is very prone to develop oral cancer. And out of 100 cases, you will find 7 to 10 people are transforming into cancer. So this is the cause of the oral submucosal fibrosis. One of the main causative factors for oral squamous carcinoma, oral submucosal fibrosis and oral squamous cell carcinoma in Asia. This betel nut. This betel nut, sometimes we have just seen that people are telling that yes, betel nut is safe, but it has got nitrous group, amino group. So this is also carcinogenic. So some people that tell that yes, I'm not chewing any tobacco. I'm chewing only betel leaf and betel nut. But this is also not safe. And we have seen that yes, this betel leaf, betel nut is the cause of oral submucosal fibrosis, and this oral submucosal fibrosis may develop cancer. 7 to 10% people are suffering from cancer from this oral submucosal fibrosis. So different varieties we have seen, and I studied extensively. Of course, WHO has their own monograph to explain about this betel nut things, and we also studied with different type of the betel nut, and we have seen the nitrous group is very potent carcinogen, and it is available in here. And this betel nut has got copper, very high amount of copper inside the betel nut. And this copper is one of the causes for development of oral submucosal fibrosis, because some regulatory method which is just inducing fibroblast to deposit inside the mucosa, and that is creating the oral submucosal fibrosis. And that may lead to cancer. 7 to 10% people are suffering from cancer from oral submucosal fibrosis. So this is the nonsense for us. So it's not that the betel nut is safe for us. It's not at all safe for us. It may create oral submucosal fibrosis. You have to understand this. So this is one of the threatening conditions for our community, as I told, that the sachet is still hanging. It's banned, but still hanging in the outlet. And we have seen a lot of the young population, the adult young, they are buying this product and they're putting this inside the mouth cavity. And they are developing oral submucosal fibrosis and other precancerous condition. So, this is the pan, traditional and long-standing habit in Southeast Asian community. You can see here there are certain things, that is all the compositions in the pan are carcinogenic. When we have started the lime, the catechu, the betel nut, and the betel vine itself is not carcinogenic, but they have when you wrap it up in combination, then all the carcinogen are going inside your mouth cavity. And if you keep on chewing this pan, that may lead to develop oral precancerous condition, and even from oral precancerous condition, if you have severe level of dysplasia, then may develop cancer. So this is not very safe. Is that punching is not also safe for us. So this is one of the pictures. We can see that how the khayni is just killing the people, and invariably they are just developing the condition which is known as oral submucosal fibrosis that may lead to cancer. So this is one of the conditions we have seen, and the lichen planus also develop associated with the chewing tobacco. This is the quick screening and education one-to-one approach we developed at Mangalore when I was working there. Just this is one of the things I have just sharing with you. And zero-style mint, a smokeless tobacco in Japan, and threatening for human lives. And this is very common. Even now in UK we have seen the Dept e-cigarette, but it's not safe. And a lot of young people, they are suffering from some sort of conditions in their mouth cavity and in their lung due to this vaping. So, and it is also very, very common in Japan, zero-style mint, and a lot of advertisements coming up. But this is not so safe. And this is also inducing some of the young people to invite the smoking habit. They start with the vape or e-cigarettes, but they transformed into cigarettes. So they start cigarette smoking. So this is not safe for us also. So in the UK, we have a strong committee and activist group to stop vaping, and a lot of outlets now stopped. And in the UK, we have seen the number of people who are smoker, they stopped, and the number of people are just reduced a lot. But unfortunately, some people, they have started vaping. That is also good for us, and this is also leading to some sort of precancerous conditions and some of the health consequences leading to the illness in the respiratory system. So the lung is also affected differently. So we are putting our effort to stop the vaping or e-cigs. You see, this is the one thing why after chewing pan, if you spit out, then red things comes, because this is oxidative reaction starts that way with the lime, catechu, and that creates that saliva become red. So this is the condition, oxidative reaction. And when you have oxidative reaction, then all the tissues inside the mouth cavity, that is mucosa, also there are certain changes happens at a DNA level. And that's also creating a risk for the human health to develop cancer. So we have to be very careful not to just continue pan chewing habit. And this is very long-standing cultural habit in this country. So, and some other regional countries also, they are doing these things, but we have to stop this sort of habit. And because we have seen that we can see the people who are suffering from, who are smoking, we can see them, but who are chewing tobacco, we cannot see them. That is the problem. So we cannot control them. So, but if you have the intervention and the education in the community, then you can stop it. So this is the killing stop. Mundas, one of the security people, they just chewing these things, and he developed oral submucosal fibrosis, and consequently, after some few the months, developed oral squamous cell carcinoma. So these are the things they develop, self-made kidney. And we have the published research on mutation and deletion of tumor suppressor genes, Coine associated. Can you just open that, the PDF file over there? Yeah, as I said, that yes, this thing, I'm showing you the picture, what is happening associated with the coine chewing or pan chewing. But if we go through the in-depth the study at molecular level, then what is happening? I can show you. And this has been studied by myself and my team in Japan. Can you open it? There is another file you can see there. Okay, you cannot do it. Okay, just you proceed on to the next slide. Then I will discuss about this paper later on. Yeah. So you can see the deletion map, because if I just read the published paper, then it will be easier for you. You see, there are, I have no mic here. Hello? Okay, so we had developed a deletion map. Hello? Can you hear me now? Yeah. We have a deletion map, and this deletion map we developed from the tissues we collected from the patients who developed oral squamous cell carcinoma associated with quinine chewing in India. Most of the patients are from Odisha. And of course, number of people using quinine in this city also, and West Bengal as largely. So we have seen there are chromosome 2q, 3p, and 21q. So loss of heterozygosity happening. So we studied, and we have these two areas are very common, the deletion. So this is D3S1007 and D3S966. This is very, very common, the loci. These areas are deleted. So this is very, very disturbing situation for us. So when we can see the map, then some of the loci and some of the genes are deleted. And the 2q is the long arm, and 3p is the short arm, and 21q is the long arm again. So this we have just we studied these three genes, and three genes are very important genes for us, and so the deletion and mutations has occurred associated with the chewing tobacco. This is one of the findings. The number of cases we have studied 26, and it's similar the situation we have seen in Japanese cases, but it is very common among the patients who are collected from the tissues collected from the patients from India associated with the chewing tobacco. So you must be very careful, and you have to inform the people that yes, the chewing tobacco or smoking is not simply developing your cancer, but it is giving some changes inside the gene, inside your at molecular level. So that should be noticed by yourself and by your colleagues. So this is one of the information we are giving to you from this study. Next slide, please. Yeah, this is the deletion and mutation of tumor suppressor genes, loss of heterozygosity. So the NIF is informative, NI is non-informative. You can see here. And normal DNA and tumor DNA. You can see this 2q, 3p, and 21q. All these areas are here, you can see clearly. And this is the things we have done it at one of the laboratory in Japan, Tokyo Dental College. Thank you. Next, please. The combination, the same thing, combinedly I am showing to you. Next, please. Of course, the angiogenesis and LVD, the lymphangiectasis is one of the things we have compared to the Japanese patients. We have seen the patients who are from Japan, suffering from India, suffering from oral squamous cell carcinoma. They have the MVBD is very high. So who are having the chewing tobacco more than 15 years, so 65.5. 65.6%. So the MBD, this is very disturbing situation for us. Even both chewing and smoking, they have more higher. So more high things are happening among this MBD, that is 71.8% here. And at the same time, LVD also, we can see here, 45.5%. So in female, this is the male, but in female it is less among these cases. So when you compare the mean microvessels density and mean lymphatic vessel density, we have seen that the people who are suffering from cancer or precancerous condition associated with the chewing tobacco and smoking, so they have a high MBD and LVD. So this is the one of the detector we can just see and can compare between these two type of the conditions, the patients from Japan and from India. Thank you. Next slide, please. This is how it does work, angiogenesis. So this is the little detail here. I cannot explain all the detail here. And this is the things it is happening with for angiogenesis. This is the picture we can just see here how it happens. Next, please. Next, please. So we have to just control and to combat this situation, prevention and control. We have the model for knowledge, attitude, practice, KAP. So this is tested model we piloted, and based on community development for health development, this concept we used it. And of course, we always consider the context and culture of the society and the population. That way, we use this sort of the CAP indices to intervene. And so we are the provider Center for Oral Disease Prevention and Control. So we developed it at the Department of Oral Biology and Genomic Studies at ABC Dental School, NIT University, Mangalore, at B4. So this study also published, and this is working, still working. But this sort of the knowledge attitude practice intervention is not existing in this state. So possibly one of the measures we have to take to use this CAP intervention in this city to prevent and control the cancer, not only in this city, in this state, starting with some certain places like SNU. If they start this sort of things, possibly it will help develop certain things to prevent and control, and it will be replicated elsewhere in this state. Next, please. This is in detail. I published this book, and we have this method. This is the guide for, it's a guidebook for based on CAPE methodology, and published in 2010, Bournemouth University, UK, and NIT University, India. Jointly, we have done it. So detail, you can find it in this book, how we can intervene with the KAP. Next, please. This is one of the Stop Tobacco session by Community Involvement Health Development. So we have developed it, and this can be started here in this state also, I believe. Next, please. The same thing, how we interact with the people, and we intervene them, and we engage them to stop tobacco. So this is one of the model we developed, and that model can be replicated elsewhere, even in this state. Next, please. The school program is very important for us. We can develop this school program, and that will also help to start tobacco chewing and smoking from the very starting of the life of the young generations. Next, please. Yeah, so replacement of the tobacco with natural antioxidant. So we educate the community or the patients individually and in a group to take their products, what they are chewing, or they are smoking, and then replacing with them the antioxidant. So this is one of the initiatives we took, and we are successful. Next, please. Yeah, this is the screening and counseling unit we developed. Next, please. Now, the cancer prevention and control strategy. So one of the things we can just take it very quickly. One of the things will be the oral cancer, skin cancer, prostate cancer, breast cancer, and cervical cancer. These cancers are very, very much preventable. If we take the appropriate measure to stop this, the 1, 2, 3, 4, 5 type of cancers, it's very easily we can introduce it, because oral cancer, skin cancer, and breast cancer are visible. So we can screen it quickly, we can advise them quickly, and we can detect and refer them to the oncologist for early detection and prevention. So this is one of the things we can do very quickly. But there are certain other things also related to oral cancer, skin cancer, prostate cancer, breast cancer, cervical cancer. There are certain AI technology also introduced nowadays to detect and screen and refer very quickly. And in the UK, we have started this one now and successful, this model. Next, please. So as I told that, yes, this is early detection, education, research, and community engagement using the KAP, NRT, AI application, vaccination, blood test for cancer screening, and symbiotic approach involving researchers and clinicians. So we have several vaccines now available, and those vaccines are very useful nowadays. But oral cancer vaccination is not that much effective until today, but human papillomavirus is one of the viruses which are associated with oral cancer. But many of the people, the scientists, they are doing their research to come up with the appropriate vaccine for oral cancer prevention. But certain blood tests, very recently introduced in the USA, in the UK, that can predict and screen some of the cancers, of them the prostate cancer, breast cancer. Now, possibly your authority will be giving the permission to use the blood testing for cancer screening in the UK. Those things is not very expensive, and this sort of measures will be introduced even in this country, and we can help develop those things in a way very conveniently if we have appropriate host to take up this sort of initiative. Next, please. So now, phase-wise implementation, what I propose, phase one will be the established core team, finalize partnerships, and develop CPD courses curriculum. Otherwise, these things will not happen. Phase two will be we have to launch patient interest group and initiate annual cancer screening program. Phase three will be expand your international collaboration and form a consortium on genetic epidemiology. And phase four will be scale up preventive strategies and community-based research across Bengal. So these are the generic outline how we can approach. It's not very much expensive things to start, but we have willpower and we have the policy in place. Possibly it will be difficult to start this sort of things. Even with SNU, it is possible. Next, please. So another thing we have just started, but it's very difficult to take the salivary samples from this country to just send it to Japan. This is non-invasive biomarkers we are using, salivary metabolites. So this metabolomics is very important things. We are just introduced and we are just trying to validate with the regression model. So we need some more samples to study this one. And we are just doing this study with the Japanese collaborators at Yamata University. Next, please. So salivary metabolimics, a pilot study for the salivary biomarker. For validation study of a biomarker of oral squamous cell carcinoma and potentially oral premalignant lesions. And multicentric international study. In UK, we have 2 centers. One is with my university, another is the De Montfort University. We have started already, and we have no problem to send the salivary samples to Japanese laboratory. And in Japan, we have the Yamagata University. So things are going on. But in India, we have some limitations because we have the method to study the salivary biomarker. This sort of the equipments and the facilities is not existing. We have searched. Possibly we have to search more extensively whether we can do some sort of study using the saliva in this country. Otherwise, we have to send it to Japan. But in this country, we do have some limitations and regulations not to send all these samples. And then we have a stalemate for that. And I started this, I just proposed this study to Sarojgupta Cancer Center. But as the government is not giving permission to send all the samples to Japan, so this is one of the problems we are suffering from now, to get the one of the collaborators from India. Next, please. So the aim of this study is to validate the accuracy of logistic regression model for discriminating oral squamous cell carcinoma and oral premalignant disorders using salivary metabolites. These are the things we are just doing now with the saliva. It's very non-invasive things and very, very informative and easy to implement. Next, please. So primary outcome will be the accuracy of the discrimination model or validation study for salivary screening of oral cancer, a study for non-invasive biomarker. Next, please. So all the metabolites, what we have included here are hydrophilic. So malonate, then adenosine, N-acetylneuroaminate, then 5-aminoferriate, and propioate carnitine. And the expression of the relatively more accurate. These expressions are more accurate, but we cannot quantify. This is quantitative. But we have some sort of metabolites which are strongly associated with the carcinoma. So those things we cannot include here because those are very, the method is not sensitive until today. We are working on that. So possibly in future we can do all the metabolites which are just very, very much linked with the cancer. But at this stage, we have taken this sort of, this number of metabolites to study. But still, it is not quantitative, it is qualitative, simply. So we are the collaborators, Japanese side, Professor Sugimoto and Shigio Ichikawa from Yamagata University, and from UK side, myself, and Professor Martin Goodbell, and from India, still we are in progress. If we had the permission, possibly we can send the samples, a part of samples to Japanese laboratory, and if we can develop certain sort of the infrastructural facilities in this country, possibly most of the samples we can study here to get an outcome out of this thing. So the fund is JSPS and Japanese government, Ministry of Education, Culture, Sports, Science and Technology mixed. So this fund was, we just got jointly with the Yamagata University. Next, please. So as I told that, yes, these are the more relevant, but assay is very qualitative comparing to the previous group of metabolites also. So this is, as I told that these metabolites are carcinogenesis promoting. So these are very, very important for us to study in future. But initially, we'll do the other type of hydrophilic metabolites, and it's ongoing. And the purpose of this study is to develop a biomarker from the saliva, and which is very much non-invasive. So you not just prick the skin to just draw the blood. Only you have to take the saliva and do the study, and that is very very much important for us and very convenient and less expensive. But the laboratory facilities, as I said, that is not existing here, so we have to send all the samples to Japan. Some of the samples we can do in UK, but from India we cannot send to any other countries, including UK. So we have to get the facilities here. And if we have the fund, possibly we can continue the study and do the rest of the work or entire work of assaying the metabolites in this country. Next, please. So the method we use, the capillary electrophoresis mass spectroscopy, CMS. This is the method. So this particular CMS method we cannot see in this country. I have checked with some of the friends. Possibly some of you here can, my colleagues here, if you have any information, possibly we can just make a link with them and we can try to just get the facilities if they do have. Anyway, if the molecular specific assay is established, that is the antibody plus ELISA, the list of biomarkers makes better sense. But using this untargeted based mass spectroscopy to analyze samples will be okay at this stage. But it's not a quantitative measurement of the specific metabolites, but rather identifying the metabolites of our concern. Usually it is difficult to measure specific metabolites, but if any of these experts have a thought on it, will be explored. So this is the, one of the opinion and one of the arts to you people, my colleagues, the scientists here, if you have capillary electrophoresis mass spectroscopy, possibly we can continue this study in this country. Of course, we'll make a collaboration with the Japanese university, and we'll try to get the fund from there by writing the grant application. And we are in the process. But these things will be going for your the validation process, the accuracy process, but with larger number of samples, salivary samples. As India has got a very high number of the oral cancer, precancerous conditions compared to Japan. Japan is only 2 to 3% people are suffering from oral cancer. But in India, some places it is 35%. If we take the head neck area, then it will be 45% of all cancers. This is a very disturbing situation in this country. So anyway, if we can just get this metabolite study done in this country, then it will be good for us to just come out with the non-invasive assay method and to establish a biomarker from the saliva. And we just have a proposal to Shorji Gupta Cancer Center Research Institute, and Dr. Sushanto Roy Chaudhary is here, and he also just introduced, he screened the proposal, and also he gave the permission to go ahead. But any permission to send the samples to Japan or elsewhere was not very positive for us, so we halted collecting the samples from there also. But some other places we are trying, and we are finding out the way how we can go ahead with this project. Next, please.
Is available more than 20 institutes in India.
Really?
Including Indian Institute of Chemical Biology, Calcutta.
Good.
IIT Kharagpur.
Nirma University.
IIT Calcutta.
Okay.
All the institutes, they have the CA.
Yeah,
We'll
we
explore.
Each instrument. So don't worry. You can
Okay,
take
okay.
20 in India.
Then that will be good for us. I'll just get back to you and discuss about it.
Dr. Chaudhary, you have 5 more minutes.
Really? Okay. Thank you, please. Next, please. Okay. So oral cancer stem cell versus mesenchymal stem cells, possibly yet to go, a lot of study to do, but I'll not explain all these things about the stem cell, cancer stem cell or mesenchymal stem cells, because these things are already explained by professor of my colleagues here from Japan University. And then, but still we have some problem with the oral cancer stem cell, because the number of cells are very very low, we can just detect from the cancer cells, so from the cancer tissues to collect the oral cancer stem cells. But mesenchymal stem cells also very good to understand, because this will give the prognosis and then regeneration of the tissues if we can establish these MSC things. But oral cancer stem cell is very important for us to understand the responses of the treatment and the progression of the disease. So that is very important to understand for us. But a lot of work to do in terms of oral cancer study. Thank you, please. Okay, so this is stem cell therapy involves two main approaches. As I told that, yes, the targeting oral cancer stem cells to inhibit tumor growth and recurrence mainly. But as I told that, oral squamous cell carcinoma, from there if you can try to get the cancer stem cell, the amount of the cancer stem cell is not very good that way. The low number of the cancer stem cells we can collect from the cancer tissues, especially the oral cancer tissues. And of course, this is very important area to study because it inhibits the tumor growth and recurrences. And but using the mesenchymal stem cell MSCs for tissue reconstruction and to deliver therapeutic agent, very important for us also to understand the MSCs. But again, there are some problem with the MSCs. So this mesenchymal stem cell has got some problem in terms of the delivered therapeutic agents and tissue reconstruction things. It has some limitations. Possibly we have to overcome those things also. The expert is here, so he will have further the thought on it, and we can go ahead with this sort of study in future. Next, please. So we have time.
2 minutes more.
2 minutes, okay. Quickly, I'll finish it. So yeah, of course, the potential drawback I explained too. It's not that much problem, but research is always giving us the path to understand and to proceed. We'll do that. Next, please. Of course, the AI technology in cancer, and I explained to you about it. So this is promising area and yet to go. And in Europe and UK, we started AI technology very aggressively. And possibly in cancer detection and treatment, AI technology is taking very promising role today, especially in the UK. We have, because UK has a huge fund for NHS, and that help us to develop certain things for the patients. So AI technology is just very common in the UK. Next, please. Of course, there are several type of things are just incorporated in the cancer diagnosis treatment using the AI technology. One is generative AI, and another is predictive AI, large language model, LLM, and explainable AI. So these are the things specifically we explain these things. Possibly I cannot explain to you to grab all these things to put in your head. But AI technology has got huge potential to screen and make very good progress with the treatment facilities so that it will be useful for us, and started in Europe and UK. Possibly we can extend this sort of AI technology in this country, and starting with the West Bengal. Next, please. Oh yeah, this is the complicated things we are just depicted here, but the same thing what I explained is explained in this in a pictorial expression. Thank you, please. Next, please.
Dr. Chaudhary, please, can you wrap up things now?
Okay. I think I have explained all these things about this thing, and we have some things we'll do in future. Next, please. Yeah, I explained this thing. Next, please. Yeah, so the images like MRI and CT scans also is here we are doing. Next, please. Yeah, please. Oh, yeah. Please. Yeah. That's the things we'll do. Thank you very much. Thank you. I think I cannot explain those published paper that the tumor suppressor gene things, they can propagate. That's all. Thank you.
Thank you, Dr. Chaudhary, for your excellent talk. Due to time restriction and, one or two questions we can take. Any questions from students? If there is no question, I want to
please.
maybe two small things. Number one is that when you discussed everything, so we all know that the oral cancer, a percentage of oral cancer that is due to the papilloma virus. So do you think that the oral hygiene is playing much more important role in India than maybe the UK or US or Japan for that matter? Because if there is infection of papilloma virus, what you haven't taken into consideration while talking, so that can be, maybe that is playing a part in this whole game.
Yeah.
And number two is that I understand that in India, the percentage of oral cancer is much more than UK, but considering the UK statistics, in 2024, around 10,000 cases were reported, while in 2025, already it exceeded 12,000. So why the rate is increasing? And third, to adjust to this, make you, inform you that the oral cancer stem cell, there are lots of lakhs and lakhs of papers are coming nowadays, because it doesn't matter whether its percentage is less, because it is maybe 1 to 2% in each tissue, but it doesn't matter, because if you can take the biomarkers and then you can just target them. So for that, number is not that important.
Yeah, I understand.
Yes.
So in terms of the first question, possibly, that the oral hygiene has got a role to just keep the viruses inside mouth cavity. So if the poor oral hygiene, then the number of the viruses will be more inside the mouth cavity. And there are some reports already published in this way. And
in UK also it is increasing. So what is your comment on that?
so now you see the causative factors, among the causative factors of oral cancer, the papilloma virus is number one. There are a number of different varieties of papilloma viruses. I think 13 and 16 is the more important thing, viruses. But why it's increasing, we don't know. But some studies, as you told, that the poor oral hygiene is one of the causes to just get
or
the
maybe
more
tobacco, smoking is much more in UK or what?
No, it stopped.
But it is increasing also there.
It decreased a lot, but vaping is increasing.
Yeah.
The e-cigarette is increasing.
And also, while I liked your part of awareness, giving awareness. So while giving awareness, I think we have to also consider that actually the pan pata is anti-cancer. So we should not avoid taking pan leaves, but maybe not mixing with any betel nut or anything, because betel nut contains carycolin, and that is very much highly carcinogenic. So pan pata is very good actually.
Only betel leaf.
Leaf is very good. It has
only
anti-cancer
leaf.
compounds
Nothing
also.
else. Yeah.
So if there is no more question, please give him a big, So excellent lecture. And I also thank the SNU family for giving me the chance to share this session, and special thanks always to our beloved Professor.
It is my privilege now to invite Professor Tania Das to felicitate our honored speaker, Professor Chittaranjan Chaudhary. May I now request our Pro-Chancellor, Professor Dhrubajot. May I now request our Pro-Chancellor, Professor Dhrubajoti Chattopadhyay, sir, to felicitate our Honorable Session Chair, Professor Tania Das. I invite our distinguished guests and all participants for a short tea session now.
because for the many years researches are going on in the field of cancer biology, how to detect cancer and how to get remedy for that. But we all understand that still today cancer is considered as a deadly disease. So I understand that maybe 30-35 years ago, the discovery of cancer stem cells, which we consider as the major root behind this deadly disease. That is the cause behind cancer development, cancer migration, metastasis, and also cancer relapse. So discovery of those cancer stem cells, I understand that that is a very big breakthrough. I should rather say the paradigm in cancer research or idea about cancer has been completely changed after the discovery of cancer stem cells, because now we understand that by targeting cancer cells themselves might not give the real remedy from cancer, but targeting these cancer stem cells, which are actually the origin of cancer cells. So by targeting them can in near future give a real answer to this disease.
What new technologies showcased here stand out as a game changer for early detection or treatment?
This is also a very good question. I don't think that single technology can do that, because first of all, cancer is a very complicated disease. It is not a single disease, actually combination of multiple disease, multiple disorders, and so not a single technology can detect everything. But first of all, the idea about personalized medicine, that has actually that has done really a very good job, because previously we didn't know that the breast cancer, for example, breast cancer of someone like X may not be same as the breast cancer of someone like Y. So it is very much personal to whom it has happened, her or his or her genetic makeup, and many other factors, molecular crosstalks, many things are on those the cancer and its remedy is dependent. And so it is very much personal. So this concept is one of those, and another is surely AI guided, detection, and then technology. All these things I should appreciate that. And these things are in my understanding, these are the major factors. And with that, we should also consider the awareness. Yeah. My designation. I am Professor Tania Das. I am Emeritus Scientist at Bose Institute. Thank you so much. Thank you.
Hello. To ask the Hello. So we are starting the scientific session after the tea break. So I would request Professor Gauri Shankar to please come up and take the chair to chair the session. Our speakers are Shayani Dasgupta, Dr. Shayani Dasgupta, and Dr. Damayanti Ghosh. Sir, please.
Hello, hello. The audiences are taking tea, I guess. Most of the students are outside. So shall we start? Yeah. I always love to come to SNU. That because of my guru, Professor Dhruvochoti Chattopadhyay, from whom I learned the research. Sir, pranam. It's my pleasure to chair a session where two speakers, I would say two young speakers are going to talk to their latest research. One is Dr. Shayani Dasgupta, the Assistant Professor of Microbiology, and the next speaker is Dr. Damayanti Das Ghosh, also Assistant Professor in the Department of Allied Health Science of Sister Nivedita University. Damayanti was an alumni of Calcutta University Biochemistry Department and did his PhD from the Chittaranjan National Cancer Hospital, then did his her postdoctoral work. Shayani. Yeah, Shayani. Sorry. Dr. Shayani did her master and PhD from the Calcutta University and then did her postdoctoral research in the MD Anderson Cancer Center, USA. His research is hard. I'm sorry. Her research interest is in the intersection of cancer biology, tumor immunology, vascular biology, and metabolomics. All of the subjects are from my heart. So she is now trying to develop our independent laboratory in SNU, and she is going to talk about the unsung architect of cancer, why vascular cell matters. Shayani, please.
Thank you, sir, for my introduction. Good afternoon, everyone. Good afternoon, all the dignitaries here, my professors. So, Tania ma'am, SRC sir, and Gauri Shankar sir, they are all my mentors. When I was in PhD, I used to go to listen to their talk in various seminars and conferences. So, and also good afternoon to my students. So today, I will take you onto a journey in tumor microenvironment. But this journey is not the usual journey where we will target tumor cells or immune cells. Today I will focus on the unsung architects who shape the tumor microenvironment slowly beneath the surfaces, and these are vascular cells. So vascular cells, the pointer? Okay. She said this. pointer or the slide changer? Slide changer here. Slide change হচ্ছে না?
না এটা তো মাইক্রোফোন
হ্যাঁ হ্যাঁ
নেক্সট স্লাইড করে ওখানে বলো
আচ্ছা okay. So the vascular Okay, carry on. Sorry for the interruption. So cancer cells, vascular cell doesn't mean it will only bring oxygen or nutrients into the tumor. They play various critical role in metastasis, in like immune invasion and evasion, and also play a very critical role in making decisions how the immunotherapies or other therapies will work on the tumor. So today I will start with my presentation entitled The Unsung Architects of Cancer, Why Vascular Cells Matter. So the story begins in 1961, where Dr. Judah Folkman noticed that the tumors cannot grow without recruiting or formation of new blood vessels. So later in 1971, he published his hypothesis that tumor cells can only grow to 1 to 2 millimeter cube without any blood vessels recruitment. So to grow, they need the formations or the recruitment of blood vessels or angiogenesis. This idea gave the birth of anti-angiogenic therapy, and this opens the door to understand the vascular biology and to understand the vascular biology and think that vascular cells are new, druggable, and dynamic systems. So years later, finally in the year 2000, the angiogenesis defined as an important hallmarks of cancer, which was published in a review work written by Hanahan and Wenberg. I can use this mic, right? Yeah? So the two most important instruments of vascular cells are endothelial cells and pericytes. So endothelial cells are like bricks. They make the tubes. And pericytes, they are like the cementing material. They wrap around the blood vessels to maintain their integrity, architecture, and also maintain the blood fluid. So in normal tissue, the blood vessels are, you can see that the blood vessels are highly organized, tight, beautifully structured. But why? Because the pericytes are attached to the endothelial cells, making it tight and make the blood flow easy. But where in tumor blood vessels, everything is broken. It looks like a city after an earthquake. So everything is broken, pipes are leaking out. So what happened in tumor blood vessels, tumor microenvironment? The pericytes lose its associations, so that the vascular vessels becomes very disorganized, leaky, tortuous, and as a result, what happens? The oxygen delivery becomes limited, the therapeutics, the drug delivery becomes limited, the pressure, interstitial high pressure becomes high. Okay. So from this, I started to see how vascular cells and tumor growth are interrelated. So to study this, first I collected human patient samples of infiltrating ductal carcinoma. The patient samples were taken from Chittaranjan National Cancer Institute, where I did my PhD. So I took this tumor, did a tumor sections, and then stained it with CD31. CD31 is the marker of endothelial cells. So before that, I would say that after collecting the tumors, I classified them on the basis of the tumor size. That is from T1, T2, T3, T4. So T1 is the tumor which are less than 2 centimeter. T2 are the one which are greater than 2 centimeter but less than 5 centimeter. T3 are the ones which are greater than 5 centimeter. T4 are those tumors which are of any size, but they have grown into the chest wall or skin. So after taking the tumor sections of these patients, I stained with CD31 and see that as the tumor grows, the expression of CD31 is also increasing. You can see here in T4, the field is become green. The field has become overall green, which means the endothelial cells are huge in number, and see that they are not in a particular, they are not following any particular organized. They are not organized. They are all over leaky. They are spilled over the field, which means that these vasculatures are extremely disorganized or extremely leaky. So we see that as tumor size increases, expression of CD31 increases. So what about the pericytes? So next, to study the pericytes, I isolated pericytes from these tumor samples. So how I isolated? I isolated a population which is CD31 negative, CD45 negative, PDGF receptor beta positive, and NG2 positive. So on this, I studied the expression, I checked the expression of RGS5. So RGS5 is a marker of angiogenic pericytes, where NG2 is a marker of normal pericytes. So as we see that, as the tumor size increases, the expression of RGS5 NG2 double positive, which means angiogenic pericytes, the population also increases with the tumor size. So we can say that from here that vasculature or tumor vasculature abnormalities is directly or is correlated with the tumor growth. So higher the number of tumor cells, so we can see the higher the abnormal CD31 and pericytes within the tumor microenvironment. So you have seen the expression of RGS5, I have checked. So RGS5 is a very peculiar molecule. Why? Because when it is outside the tumor microenvironment, the literature says that it induces cell death. It induces apoptosis of various cell lines, like endothelial cell lines or even ovarian or lung cancer cell lines. But when we see that the expression of RGS5 is highly upregulated within the tumor microenvironment, these are associated with poor prognosis. Poor prognosis, abnormal vasculature, decreased response to immunotherapies, tumor angiogenesis, and metastasis. So it's like, why this is the dual role of this RGS5. So next, I targeted or focused my study on how this tumor pericytes, like RGS5 expressing tumor pericytes, have role in angiogenesis and tumor microenvironment within the tumor microenvironment, and also on another aspect, I started studying that how it can immunomodulate the tumor microenvironment, whether they have any role in immunomodulation within the tumor microenvironment, and then foster tumor growth. So look into the RGS5 canonical pathway. So RGS5, so we know that GPCR is a 7-membrane receptor. So once G protein is usually in this inactive form, binds to the GPCR. So when it gets stimulated, GPCR has 3 subunits: G alpha, G beta, and G gamma. So once the GPCR have any signal, it gets activated, and the G protein and G beta gamma subunits dissociates. Now the G alpha subunits is attached to GTP. So each of them induce various signals. So among them the signal is PI3 kinase pathway, where G beta gamma subunit can activate PI3 kinase pathway. So once the signal is turned on, you have to terminate the signal. So there comes RGS5. RGS5 is regulator of G protein signaling 5. RGS5 binds to the G alpha subunit of G protein. So once it binds to the G alpha subunit, it hydrolyzes the GTP. So once the GTP is hydrolyzes, the G alpha subunit is now bound to GDP. Now once it is GDP, it again associates with the G beta gamma subunits and become inactive. So when G alpha protein, so it's like when the term, this signal is on, when RGS5 is not bound, so the PI3 kinase AKT survival pathway is on. When RGS5 is bound to GTP, it will stop the survival pathway, and it will promote apoptosis or cellular death within the cell. So next I was studying that what is the role of RGS5, whether the apoptotic status of RGS5 expressing pericytes within the tumor microenvironment. So to check that, to stain the RGS5 high pericytes isolated from the tumor with annexin 5 and caspase 3. These are the marker for apoptosis. And I see that as the tumor size increases, the expression of RGS5 increases, but the expression of annexin 5 and cleaved caspase, they are decreasing. which means within tumor microenvironment, these RGS5 high pericytes, they are not undergoing any sort of cell death. Okay, nice. So with RGS, so they are inversely proportional. To mimic this result, we inoculated tumor in murine model, inoculated this mice B16 with B16 F10 mice, isolated the pericytes from at different stages. So at different stages means we are on the basis of the tumor stage, we categorized the tumor like early, middle, and late. And from there, I isolated the pericytes. A similar trend was observed, like with the increasing in the RGS5 expression as the tumor size increases from the early to late, the expression of RGS5 increases, but the expression of annexin 5 and caspase 3 decline. So it was okay, the RGS5 is not promoting apoptosis. So what happens in other normal physiological organs? So next I isolated the kidney, isolated pericytes, RGS5 high pericytes from murine to kidney. But it was like the result is different. Like this RGS5 high pericytes promote apoptosis. It's like showing 44% of annexin 5 positivity in RGS5 in these pericytes. So, but what happens in B16 melanoma and other type of cancer, Lewis lung carcinoma? So these RGS5 pericytes again showed decline apoptosis. So it's like I was baffled, like why this is happening? What happened? What is there? What is the difference between this RGS5 and other RGS5 in normal tissues and cancer tumor tissues? So I shifted to check whether this is the problem in RGS5 or not. So I shifted into the in vitro system. So there I overexpressed RGS5 in low pericytes. So when I overexpressed RGS5 in low pericytes, again I found that when RGS5 is high, the annexin positivity is high. But when you silence the RGS5 with siRNA, the annexin 5 positivity decreased. Okay, nice. What happened? So it's like, I was like, anything in the tumor microenvironment must be there, which is regulating this dual behavior. So next I exposed those RGS5 high pericytes, in vitro pericytes, to tumor lysate. So when I exposed them to tumor lysate, the expression of annexin 5 positivity in these pericytes declined to 6%. Okay, so it must be in the tumor microenvironment. This tumor milieu have something. Now, next, my target was to find which is the factor. What is the factor behind this dual behavior? So I exposed this RGS5 in vitro pericytes to different tumor lysates, and then neutralize this lysate with a panel of different cytokines. So interestingly, I found that when you neutralize those tumor lysate with TGF-beta, the apoptosis of this RGS5 high pericytes is restored. It's again 35%. So if you add recombinant TGF-beta to it, it get declined to around 1%. So it's like, so it's to me it's interesting why this TGF-beta suddenly. So it's next I checked that what happened. So that next you can see here. So when there is low pericyte, the RGS5 pericyte is not expressing it is the RGS5 low pericytes. The pericytes are not expressing RGS5, and you treat them with TGF-beta. The expression of annexin 5, the cells are apoptotic. Annexin 5 is high, the white one. So if you normal pericytes you overexpress RGS5, then also the expression of annexin 5 is high. But when you have both in like RGS5 high pericytes exposed to recombinant TGF-beta, the annexin 5 positivity declined drastically. Okay. So like next one, what is the mechanism behind it? So why this is happening? So like to study the mechanism, so like whether it's like RGS5 having any different role, or what is the alternate mechanism then? So next, in both in vivo and in vitro culture, what I did, I pulled down, I took the lysate, pulled down with RGS5, and did a panel of Western blotting. And I found that RGS5, which was supposed to bind with G-alpha inhibitor in late tumors or in presence of TGF-beta, it doesn't. It is not binding with the G-alpha inhibitor anymore. Okay, when it's not binding with the G-alpha inhibitor, both in the late tumors and in presence of TGF-beta. So what happens to the G-beta gamma? So G-beta gamma subunits, it remains bind with the catalytic subunit of PI3 kinase, that is P110 gamma, which already said by Dr. Senu, that PI3Kg, P110 gamma. Okay, so if this G-beta gamma is bound to PI3 kinase, so it means that PI3 kinase AKT survival pathway is still on. So now look into the canonical pathway for TGF-beta. So TGF-beta normally binds to SMAD, and this SMAD and regulatory SMAD, and then bind to co-SMAD, which translocates into the nucleus and promote apoptosis, promote transcription of various apoptotic genes. So what happened, we see that here, a very interesting story, that RGS5, now not only, it's not binding with the G-alpha inhibitor, but rather it binding to the SMAD2 of TGF-beta. SMAD, when it's binding to both in vitro conditions and in vivo conditions. So why this thing happened? So when we do our further bioinformatic analysis, we found that this SMAD2 share a common binding region with SMAD4 and also with RGS5. So this SMAD2 has higher binding affinity for RGS5, as they share larger interface area with it. And next we found that this SMAD2, this RGS5 in presence of recombinant TGF-beta, which was RGS5 is normally a cytoplasmic protein, but it's translocating into the nucleus, and when in presence of TGF-beta, and I found that not only it's translocating, it's co-localizing with the SMAD2. So what happened here? TGF-beta now, in presence of TGF-beta, because cancer cell has a lot of TGF-beta within the tumor microenvironment, this SMAD2 binds with RGS5, both together translocate into the nucleus, but here the SMAD, which was about to bind to the SMAD binding element to promote transcription of apoptotic genes, it's not happening. Now RGS5 prevents it binding, which we confirmed by chromatin immunoprecipitation assay, both in vitro and in vivo systems. So RGS5 complex doesn't bind. So as a result, so all the survival protein, anti-apoptotic protein upregulates, and it downregulates the pro-apoptotic proteins, both in vitro cultures and in vivo, and in pure patient samples. So whether this RGS5 and TGF-beta is important for angiogenesis or not. So next, we did a very interesting experiment where I inoculated the mice with tumors, and after they reach when all the mice reach an approximate tumor size, I treat them with this component LY210, which is a TGF-beta receptor inhibitor, control TGF-beta and RGS5 siRNA, TGF-beta siRNA alone, RGS5 siRNA alone, and both. So interestingly, after treatment, I treated them for 3 times in an interval at day 15, day 9, 18, and 21. I found that this, so when there is no treatment, the adjacent blood vessel to the tumor is very thick. So I'm treating with this TGF-beta receptor inhibitor, it's thinner. Control is nothing more happens. Only with TGF-beta siRNA and RGS5 siRNA, the blood vessels thins. But drastically, when you see that when they were treated with both RGS5 and TGF-beta, the blood vessels is almost diminished. It's drastically reduced. So even if you compare with the palpable tumor, the tumor which is growing, they also have adjacent blood vessels draining into the tumor. But this is not happening when RGS5 and TGF-beta siRNA was treated together. This part was published in in 2021.
Wrap up.
So next, I neutralized the TGF-beta, where we thought that TGF-beta neutralization was promoting apoptosis of RGS5. So what happened to the RGS5 low pericytes? So I found that when it's promoting tumor, I isolated this RGS5 pericytes from these two groups when was treated with PBS or TGF-beta neutralization. I found that G-alpha inhibitor regains back or restore its binding with RGS5. As a result, all the caspases were upregulated. Interestingly, I found that when I isolated this RGS5 and positive and RGS5 low pericytes from these mice, we have found that the expression of angiopoietin is increased in RGS5 low or RGS5 negative pericytes, and the endothelial cells, again, the endothelial cells showed decreased angiopoietin 2 expression, and Tie2 receptor expression was also increased a little bit. So what does it mean? This means change. It means that when the this Tie2 receptor binds with angiopoietin 1 and angiopoietin 2, both, but when it's angiopoietin 1 binds to angiopoietin 2, when the angiopoietin 1 binds with the Tie2 receptor on endothelial cell, it means vessel stabilization. Whereas when it's angiopoietin 2 binds with the Tie2 receptor, it means vessel sprouting. So here we find that after tumor sectioning of these two groups, I found that in PBS you can see that the CD31, the endothelial cells are very leaky. They are not organized. Whereas on TGF-beta neutralization, I found most of the fields, the endothelial cells are more more proper and integrated, and they are forming a vessel-like structure within this upon TGF-beta neutralization. So I found that TGF-beta normalization normalizes tumor vasculature by promoting the attachment of pericytes via this Tie2 angiopoietin 1 axis. This is the overall summary which I've already explained. I'm not going through it because of the time restriction. Next. So I conclude there that cancer is not a disease of cells that growing out of control, but is a disease where conversation gone wrong. So it's like vascular cells play a major role in conversing, where the establishing the communication with the tumor cells, immune cells. So we cannot, it's no more a bystander. We should consider this as an important factor, because many of the drug therapies, chemotherapies, immunotherapies depends on what is the status of the vascular cells. How vascular cells are, how vascular cell will carry the therapies from the peripheral to the tumor microenvironment or within the tumor cells, how it will target, and how the immune cells are egressing out from the tumor to the dendritic cells. All depends on the structure of vessels. So we cannot neglect it. We have to consider it, and then we can think about further potential therapeutics, right? So understanding this vascular immune interplay is very much needed to normalize blood vessels and improve immune therapies. So at the end, I would acknowledge all my PhD supervisors, my lab mates in Chittaranjan, my postdoctoral supervisors. Now I would acknowledge Pro-Chancellor Sir Dr. Drupajyoti Chattopadhyay and our Dean for giving me the stage to present my data here, and also the Assistant Director of Microbiology and Biotechnology. And I extend my thanks to all my colleagues here. Thank you so much.
Thank you, Sayani. Since the time
Thank you, and I'm happy to
constrained,
take any questions.
Yeah, there are maybe one or two questions from the audience, particularly from the backbenchers, the students.
One important layer in angiogenesis is BGF.
Yes, sir.
So how that is in your story, how that
So VGF is mostly from the endothelial cells. So I have not checked the VGF here. So, but here, what I saw that PDGF receptor in pericytes, in the context of pericytes, PDGF receptor beta, TGF receptors, they play a much bigger role in angiogenesis. So VGF is on the endothelial cells.
So I want to make a comment
Sure.
on this answer that it has been established also that cancer stem cells also can make huge amount of VGF.
Yes.
And I have a question also that because it is binding to the alpha subunit and neutralizing it is binding to alpha subunit.
It should bind to the alpha subunit, but in tumor microenvironment, it is not.
So my question is, you are considering only the trimeric G protein or high molecular weight dimeric G protein also?
Here I only consider the trimeric G proteins, G alpha.
So I have a very small question. Since this RG5, it binds to the GI alpha. That means the cyclic AMP level goes up.
Yes.
because it's an inhibitory G protein. So does it has any consequence on the expression of either PDGF or VEGF in endothelial cells? Because pericytes are the next door neighbor in the endothelial cell.
So
That's the most sensitive cell in any cases, particularly in tumor microenvironment.
So that's the thing, sir. I didn't the very first in my first study. So I only looked into the pericytes. So in that context, I didn't look into the endothelium.
Because pericytes cannot exist alone. They need the endothelial cell for their support.
Yes.
So both are dependent to each other. So
Yes.
you must also look at the endothelial cell, how pericytes control endothelial cell or in the vice versa. That's very important because they cannot exist or work alone.
So yeah, they didn't work alone. But when I see that, so when it's like working here, like what I said that TGF. So what I did, I only checked the pericytes. So isolated the pericytes. Yes, but when we have the spatial organization, like it depends on the spatial orientation. So I didn't count on that. How it's like how the spatial arrangements, how pericytes will like having much more effect in directly in the communication, which I want to look into it. I didn't see.
But since you are working in tumor microenvironment, it is very important to see whether they are working in a cooperative way or together. Because in microenvironment, nothing works alone.
Yes, nothing works alone.
Yeah.
Yes. So hypoxia is a role because RGS5 is inducible by hypoxia, HIF1 alpha. So the RGS5, it's like I haven't shown. It's there in my article that if like RGS5 only expressed when HIF1 alpha is sits in the promoter. So hypoxia, obviously there must be a role because hypoxia is playing a role there in upregulating RGS5. So oxygen supply didn't check how it changed, but I checked the hypoxia thing.
Okay, thank you, Sani.
So one question.
One question.
I do have a question that actually two, but first one will be how does the vascular normalization improves immunotherapy?
So how does vascular normalization improves immunotherapy? It's a good question because when the pericytes and everything are aligned, if you treat the cells, if you treat with any drug, so the cells or drugs, it will directly go into the tumor niche. So if this is disorganized, the drug penetration, it will dissociate further. It will dissociate faster. So you can see, because in my postdoc I have seen that if I have the picture, I can show it. So I was about to say that, but it's not. I didn't include it here. So I have seen that when it's like that when blood vessels are growing into the tumor, so there are some regions which are leaky and there are some regions which are intact. So with what I want that drug to be there for a longer time, so that it penetrates slowly into the tumor microenvironment. But what happens if the blood vessels are not organized, if the pericytes are not organized, they are leaky, it will come out very fast. And if it's come out very fast, it will also clear out very fast. So the drug doesn't remain for a longer time into the tumor microenvironment, and it's not functioning properly. So that vascular normalization is much needed for better therapies to work.
Okay, thank you, Sani. Our next speaker is Damayanti, Dr. Okay, or maybe after the second talk we can do it together. Yeah. It's my pleasure to invite Dr. Damayanti Das-Ghosh, Assistant Professor, Department of Allied Health Science of the SNU. She is also the scientist in Department of Basic and Translational Research. Oh, she was. And the Technical Director of Molecular Diagnostics Lab at Saroj Gupta Cancer Hospital. Sani did her master from the Calcutta University and did the PhD from Indian Statistical Institute. Yeah. So her interest is in to understand the influence of some host immunological and viral factors in pathogenic HPV and H16 and 18 pathogenesis in cervical cancer. Thank you, Sami. Yeah. And her title of the talk is The Curious Case of P53 in Gynecological Cancer.
So I'll just take this mic. Thank you, sir, for the kind words. Actually, that was my interest when I was doing my PhD, especially HPV-related cervical cancer. But later on, I got the opportunity to work in ovarian cancer and also in endometrial cancer. So that's why, and I have been also studying TP53 mutations in different gynecological cancers. So I thought of bringing up this topic and discussing it in front of this august audience, that there is actually a curious case of TP53 in gynecological cancer. So I just want to go ahead on that. Next slide, please. So the gynecological cancers are actually 5, including uterus, cervix, ovaries, vagina, and vulva. But we'll be sticking to uterus, cervix, ovaries because they are the most common ones. Next slide. Next slide, please. If we see the difference, how it has evolved from 2012 to 2022, just in 10 years, breast cancer, as we know, is no more, it has become the topmost female cancer, and cervical cancer, which was the topmost cancer, the most predominant cancer in 2012, now it's the second common. Ovarian cancer being the third. So there is this change. Keeping that in mind, we must be ready for the next changes also, which may arise because of lifestyle changes, because of our exposure to environmental factors, different things. So next slide, please. So if we compare between the Western world and us, we can see that endometrial cancer is on the high, the green one under Northern America. But in case of India, endometrial cancer is not that high because it's more hormone-related, and it's like the breast cancer milieu. It has a lifestyle factor also in it, and a hormonal factor. So maybe the ladies who are more prone to having an obese physiology may have an endometrial cancer. So that is not the scenario in our case, because in India we know that majority of the ladies are still below poverty level. I'm talking only about the ladies because I'm dealing with gynecological cancers over here. Next. So, but we don't know. The scenario may change. So those cervix, ovary, they are the topmost gynecological cancers. I have kept into the in the perspective endometrial cancer also, which is just following the trend. Next slide. That was gynecological cancers introduction. Now, the other part of my talk is guardian of the genome, P53. Majority of the students, they must be knowing that P53, as the feature, the animation or the cartoon shows that this one is standing just like a superwoman or a superhero and guiding our genome from any kind of stress. If there is a stress, then it will go for DNA repair, cell cycle arrest, and then apoptosis. And by that, it will maintain the genomic integrity. That much is known. This slide was mainly for the students. Next slide, please. Now, giving that introduction about the two aspects of my talk, gynecological cancer and P53, I'll now move into the individual cancers. Cervical cancer, which is the most frequent gynae cancer, which is coming from the reproductive system of the woman. Okay? So it is the most frequent gynae cancer in India. And it is because of a persistent infection, mainly the majoritiology is the persistent infection of sexually transmitted high-risk HPV, that is human papilloma virus. Next slide, please. Although there is around less than 10% cases which are having a P53 mutation as well, as far as this one shows. You can see the P53 mutation over there. But mainly the P53 is not mutated over here. So that raises a question, because P53, we know like it's mutated in 50% of the cancers. So why not here? Next slide. Because it's not, because for the students again, it's E6, a particular protein, which is an oncoprotein of the HPV genome. It is coded by the HPV genome, which degrades P53 either by proteasomal degradation pathway, or it inhibits P53 action by binding to the, by inhibiting the acetylation of the P53. Next slide. So in this case, in the context of cervical cancer, it's more like whether E6 is present or not, whether E6 is actually inhibiting P53 or not. And if the HPV genome is intact, then here are E6 and E7. These are the two oncogenes. So E6 is mainly related with the P53, and E7, it inhibits PRB. So let's stick to E6. E6 is actually inhibited by E2. E2 acts as an inhibitor. It's a repressor. When it is possible, if E2 is intact, that is, this virus is episomal. But if it is integrated, that is, it is disrupted, then just near E2, the disruption takes place and the host genome is inserted. I mean, the virus is inserted into the host genome. And somewhere in here lies E4. So it's in, next slide, please. So I had asked this question, that whether we can actually quantify how much of the genome is, HPV genome is episomal and how much is integrated, because by that I can actually find out whether E6 is working and whether P53 is actually being inhibited. So here, by that, this was an assay that we designed, and by comparing E7 and E4, you can see over here, because E4 is actually replaced by the cellular DNA, by comparing these two, we can actually find out the status of the genome, whether it is disrupted genome, that is, it is inserted within the host genome, or it is purely intact, that is episomal, or there is a concomitant version that is both episomal and disrupted. So in that way, if we consider more into cervical cancer, it will move more towards the HPV genome related questions. So we will be rather shifting from the P53. So next slide, please. So now, moving on to the next cancer, which will show you a completely reverse scenario. Ovarian cancer. It is the second most common cancer among Indian women. This, and mainly, it is the type 2 epithelial ovarian cancer, which is also called the high-grade serous ovarian cancer, which is the most frequent, aggressive, and the recurrent type. Next slide, please. Here, the case is completely different. You can see high-grade serous ovarian cancer has more than 90% P53 mutations. Along with that, endometrial cancer also is there with more than 40%. Next slide, please. Yes, so that was it. It is more than 90%. Next slide. So now let us look into the P53 story, because now we have a cancer where there is an entire plethora of P53 mutations, which was not present in case of cervical cancer. So in P53, regarding P53 mutation, what is the wild type doing? As we all know, wild type is actually helping in cell cycle arrest, DNA repair, apoptosis, and maintaining the genomic integrity. If there is loss of these functions, why? Because of either deletion, truncation, frame shift, loss of the function of these tumor suppressor activities, then there will be no tumor suppression. But if there is another aspect to this, that is, other than in addition to loss of its function, there can be some oncogenic functions and enhanced tumorigenic functions that come upon the protein because of the particular mutations. These are called gain of function mutations. Or very recently, it was also published, it was talked about as separation of function. Like it's different from only loss of function. So it can be either told as GOF or SOF, separation of function. So what can be the gain of function? It can be, now it's completely like tumorigenesis, proliferation, migration, metastasis, metabolism, therapeutic resistance, and so on. So from there the question came, next slide. Especially in the context of ovarian cancer, where there is a role of relapse, a very high percentage. In hybrid serous ovarian cancer, there is more than 80% relapse. So, keeping that in mind, ovarian cancer also showing a more than 90% P53 mutation, we tried to ask the question whether these mutations, they are actually driving the cancer stem cell property in the cells, because cancer stem cell properties, among different properties, there is one property also which is the chemoresistance. Here we can see, this slide I especially included for the students, that if there is a clone of, a cohort of stem cells present, even with anti-cancer drug therapy, the cancer cells die, but the cohort remain and they can grow from there into the, regrow from there into the tumor. So whether this is happening because of these mutations now, because we have talked about the mutations, it's more than 90% in ovarian cancer, and these are the hotspot mutations. We generally find these mutations in different cancers in P53. So we were talking about these now. Although there can be also stop gain and frame shift mutations in a 60-40 ratio, like more 60% missense mutations, 40% truncated mutations. Next slide. So we had also elaborated about the cancer stemness related roles of P53 in our review paper. Next slide. Now, now moving further in ovarian cancer only, I would like to discuss talking about choosing the particular mutations in P53 and what is the difference in cancer stemness. So wild type T53, because we have seen that even there are a few wild type P53 ovarian cancers also, though more than 90% are P53 mutated. So what's the difference between a wild type P53 ovarian cancer cell line and a GOF P53 mutated? Next slide. So we can see that there is a difference in the spheroid formation. There is a difference. We can see that the spheroids are more in the endogenous cell line, which is having a GOF mutant R248Q, than the wild type mutant, the wild type cell. Then we can also see that the CD44 marker, which is a CSC marker, is overexpressed in OFCAR3. Next slide, please. Next, if we do further cancer stemness related assays, here again we are looking about both the CD44, CD24 flow cytometry based assay, and we can see again that in the OFCAR3 specific cell line, the GOF specific cell line, there is a increase in the CD44, both in the CD44-24 plus population and also in the CD44-24 minus population. Next slide, please. Also for the aldifluor assay, we can see there is almost a 40% shift, shift towards the stemness positive cells, showing high aldifluor signal. Next. Next, we tried to knock down P53, and a little bit a knockdown was here in the OFCAR3 cell line, and we compared both the 82780 and OVCAR3 cell lines. And we can see that because of the knockdown, there is an effect. Here you can see from the 70% stem population, we can see there is a fall in to 50% population, but the 82780 didn't change much. It was from 27 to 33. Next slide. Now, the next level was whether there was a difference between the loss of function TP53 mutant and the gain of function TP53 mutant. So again, we can find that yes, there was a difference in the endogenous cell line. We have done this one, where we can see that there is a difference of around 20%. Next. And also in the chemoresistance assay, we have seen that there is a difference where the chemo resistance is increased in more in OVCAR3 than in SCOV3 compared to the wild type variant, that is 82780. So more towards the GOF mutant, the chemoresistance has increased. Next slide, please. Now, we have also tried to define the difference between different GOF TP53 mutations, because I had shown you earlier that there are many hotspot mutations. Next slide, please. So we can see over here, so there is just a comparison from TCGA data, where we can see the OCT4, which is also called PAU5F1, is highly expressed in the R248W expressing rather mutant R248W mutant cells, clinical samples than the other loss of function mutants. But in case of R175H, that is gone. And MIC is showing a borderline significance. Next slide, please. There was also a clinical survival, a difference in the survival of between R175H and R248. You can see over here. So different TP53 mutants are actually behaving differently when we are actually coming to the context of particular cancers. Next slide, please. Next slide. Next slide. Yeah. So my student, whom me and Professor Shushanta Roy Chaudhuri, who is sitting over here, have been guiding her through this particular project, where she is now she has taken up this 82780 cell line, and she is actually trying. She has developed isogenic cell lines through CRISPR-Cas9 technology for the TP53 null and TP53 hemizygous, though this one is a little queried still now, but null she has developed. So now we are trying to see the entire mechanism in isogenic cell lines. So because of this, this is the flow, this is the plan of the work, and we would be going further to R248Q also. We will make an isogenic cell line with R248Q also, and then compare the CSC properties. Next. So while doing so, she, next slide. Yes, so this is the plasmid that she has used, and she has actually cloned the gRNA for specific for exon 3 and exon 4 into this plasmid for CRISPR, and analyzed it with colony PCR and sequencing. Next slide, please.
You have 2 minutes.
Okay, and then she has also done, now she has transfected it into the mammalian cell line and checked for it. So this is the mammalian cell line where we can see the deletion. Next slide, please. And we are now comparing between the mammalian cell lines that she has created, H3 knockout, E4 knockout, with the wild type ones, and we can see that there is a shift in stem cells, the stem cell population, and also it is reflected in chemoresistance. Next slide, please. So now moving on into the endometrial cancer. Next slide. So this was like a pioneering report from TCGA, where they found that Pol E, this is the blue line, which is having a very good prognosis in case of endometrial cancers, compared to the red line, which is having P53 mutated cases. But then there are overlaps. And in between lies MSI and copy number low cases. So, but there is an overlap. And next slide, please. So in the overlap, we further looked into the granularity that those mutations, P53 again, we are talking specifically of P53 now. So the P53 mutations along with an overlap for poly mutations, which is written as mixed over here. What is the difference in case of loss of function and that is truncated, and the other missense mutations which are having gain of function mutations also. So we could see that only the truncated variety had that is a deleted variety had a better prognosis, but not the missense variety, even in presence of Pol E, which was taken as a gold mine, that it can actually show a very good prognosis. But even in case of a poly presence, if there is a missense variety of P53 mutation, it's going to show a worse prognosis than a truncated variety. Next slide, please. And additional to that, we again found another bit of heterogeneity, that is from the missense mutations again, we could pick out Y220C. Very strangely, this is a missense mutation, P53, but this behaved as good as poly. So here you can see this line, the violet line, the purple line is just overlapping with the orange line. So they are having the best prognosis. So this one we published in our data. Next one, in this report. And with that, thank you. Next slide. So in conclusion, I would like to say that, so this is, now that I am trying to do my independent research, so I am actually thinking in the matter of, in the contextual matters. And now I have been opportune enough to look into all the 3 cancers in gynecology. So this is how I actually am summing up now. And I'm thinking about next projects and how to go about this. That in ovarian cancer, HGSOC it portrays myriad different kinds of P53 mutations in uterine or endometrial cancer, whereas in the cervical cancer, P53 has almost no mutation, and it's because it's degraded by the HPV specific proteins. And in uterine cancer, that is endometrial cancer, we can see P53 having different roles. Not all of them are as bad as we thought. Y220C has a good prognosis, almost as good as poly, and also the truncated variety, poly was better in presence of loss of function P53, that is the truncated variety, than the missense mutated P53. Next slide, please. So with that, I'll come to the end, and I acknowledge my collaborators from Sharaj Gupta Cancer Hospital, without whom this would not have been possible. And of course, my mentors, Professor Sushanta Roy Chaudhary and Professor Shurmila Sengupta and Dr. Ashima Mukhopadhyay, with whom I have been working for on different gynecological cancer-related thought processes. And also Professor Shumin Bhattacharya from Sharaj Gupta Cancer Center, who has been our head of research and head of surgical oncology. So with that, I'll come to the end, and I thank from the bottom of my heart the SNU and for giving me this platform, and also the departments of microbiology and biotechnology for organizing such a well organized symposium for the day. Thank you. Thank you for your patience.
Thank you, Damayanti, for such a nice talk. Now the paper session is open for discussion. From the audience? Yes. Yeah.
A comment for your thought. That is, nowadays we know that in cancer cells and cancer stem cells, they are very much in plasticity. So when you are considering that P53, gain of function P53 is, you are getting more cancer stem cells, we are also getting more cancer stem cells when the cancer contains gain of function P53. But I want you to think about this. We have become old. So you think about this, that they are always in plasticity. 1 minute, it is cancer cell, next minute it is the cancer stem cell. So what is happening to the mutation status and level of P53 then? You don't have to tell me now because it is very complicated, issue. You understand my point?
Yes,
Or
ma'am.
nothing is there as cancer stem cell, nothing is there as cancer cell. They are all in plasticity. So in view of that, where does this mutation status stands?
Ma'am, we have heard about, we know that BRCA1, they have their revertant mutations. So in P53 also, whether revertant mutations are possible,
That
but then how long?
there is no paper just
Yes.
on this. So just it came to my mind that what is
Thank
going
you,
on.
ma'am.
Very good lecture.
So
Thank
if
you.
there is no questions, can I ask
Yes, sir.
you questions? So P53 is a very complicated molecule. If you have it, it's good. If you don't have it, it's good. Sometimes if you don't have it, it's bad. If you have it, it's bad. So, doing experiment with P53 needs some more brainstorming session.
Yes,
Yeah,
definitely.
because if you inhibit P53, the cell will behave in a different way. If you silence P53, it will work in a different way. If you knock down P53, it will work completely different way. In addition to the mutation, I'm not talking about the mutation, because the body has a backup system always. If you knock down P53 permanently, the cell has a P63, P73 that will take the charge of P53. So when you are knocking down P53 with CRISPR system, you have to think about the P73, whether they are doing the same work what P53 is doing. Because when you are knocking down P53, the repression of P73 is gone. So P73 will automatically, they will synthesize and going up in the cells. So at that case, if you get some results, you have to think about the P53 knockdown again. Otherwise, it will be more confusing.
Definitely, sir. I'll keep
That's
that
my
in mind.
suggestion.
Yes.
Yeah.
It has to be compared always with the other pocket proteins.
Yes.
Thank you.
Yeah. Thank you both the speakers. Both of them has done very tremendous work and very nice work. Yeah.
Thank you, sir, and thank you, Damayanti. So now it's felicitation. Damayanti, if you please can stay on stage. Yeah, and no, we will do Damayanti first because she just left the stage. I thought she's already here. Okay, so may I request Professor Chittaranjan Chaudhary, if you can please felicitate the speaker. Yeah, that we discussed with Amita. Please. Okay. Thank you, sir. Thank you, Damayanti. Now, may I call upon stage Shani Dasgupta? And may I request Professor Masaharu Seno to please felicitate the speaker. Okay. Yeah. Yeah, thank you. Thank you, Professor. Thank you, Shani. We can continue the discussion after the session. So, may I now request Dr. Arun Lahiri Majumdar to please come up on stage and felicitate Professor Gauri Shankar Shah. No, I don't want to bother Sir. Sir has some, that I know. That I know. Sir has some pain in the leg. Okay, both of you, please. Our Pro-Chancellor, Professor Drupajyoti Chattopadhyay and Dr. Arun Lahiri Majumdar. Group. Vote of thanks. Okay. Thank you, sir. Yeah, we'll go for the photograph later. So, may I now request our Assistant Director, Dr. Rajat Pal, to please deliver the vote of thanks.
Good evening, everyone. So we are at the end of today's program. It's a very good lecture session what we have, and I guess all my students have gathered lots of knowledge from all the lectures what we have listened today. So I would like to thank Professor Masaharu Seno, who has come for a long way to enlighten our students, and he has spent the whole day with all of us. I also would like to thank Professor Chitto Chaudhuri. Personally, I had an interaction with Sir, so I know that what kind of work he is actually doing. But it is a very immense pleasure for all of us that we had got a chance to listen to him. Also, I want to thank our Pro-Chancellor, Sir, Professor Drupajyoti Chattopadhyay, for spending lots of time with us, for encouraging us, and our chairpersons, Professor Tania Das, Professor Rai Chaudhuri, Professor Shah. Very big thanks to our organizing members, Dr. Shaha, Dr. Dotto, Dr. Banerjee, and Dr. Sujit Singh, all other faculty members who are here. Also, I would like to thank our two HODs, Dr. Fatima Kalkatawala and Dr. Shomshubra Thakur Chaudhuri. And a very big thanks to our Honorable Vice Chancellor, Professor Shonku Bose, for allowing us to organize this session. And as I always say, that I will not thank my students and volunteers, because rather I should encourage them. So I'm encouraging their good work, and I'm appreciating their hard work, as they always do. So, and of course, I forgot the taking name of Professor Arun Lahiri Majumdar for spending so much of time with us. And okay, also I would like to thank Dr. Amit Sarkar for taking the initiative of organizing this event. So with lots of thanks, now we are at the end of our today's program, all-day program. So in our next scientific session, again we shall meet. With that hope, we are ending And I guess Mayuri is requesting for a group photo. So can all of us be here for a group photo, please? Yeah, please.
We will gather on stage for a group photo, but before that I have an announcement for the students. After the group photo, a QR code will be shown on screen, right? And from that QR code, if you scan, within the coming week, you will get the certificate, okay? Not today itself, but you just scan it and fill up the form, right? Okay, so we can just please come up on stage for the group photo. Students can go in the backside also. Sorry. Okay.